Hello, biology students! Today we're going to be talking about a specific, special type of protein called an enzyme. We're still in our first theme, the first quarter, and we're in topic 3 still, and we're on the 3rd page of our notes. Let's begin and talk about these puzzle piece kind of proteins. So the first thing you need to know is that enzymes are also called catalysts. What? I still don't know what an enzyme is. Okay, let's talk about it. So an enzyme is a protein. It's just a certain type of protein. There's tons more types of proteins than just enzymes, but let's just talk about this kind today, right? This special type of protein is in charge of speeding up chemical reactions. That definition, speeding up chemical reactions, is the definition of a catalyst. So enzymes are catalysts because they speed up reactions, right? So enzyme and catalyst actually are synonyms. You can use them the same way. Well, let's learn how they speed up chemical reactions a little bit. They do this because they have a very specific shape. Hence why we had puzzle piece pictures, okay? So sometimes we draw it all wiggly, like wiggle puzzle piece matchups. Sometimes we draw them like almost like Pac-Man, okay? And sometimes we draw it like kind of like a key going into a lock. But either way, the thing in common with all of these pictures is there's two pieces coming together and they match almost exactly like puzzle pieces. That is so important to how an enzyme functions and speeds up a specific chemical reaction. All right, let's get talking about how this works. Some of this you might need to pause and draw some visuals coming up. Not quite yet, okay? So, an enzyme works on a reactant, okay? And that reactant that it works on, all right, or does a reaction on, is called a substrate, okay? And so this is our first drawing that I really think you need to do, and you need to label the vocab words, okay? The enzyme here is this bigger shape, okay? Kind of like a bowl that has specific edges, and those edges match up like a puzzle piece with our pink shape, the substrate. So our enzyme is able to do a chemical reaction, and that substrate is going to go through said chemical reaction, all right? That's what the enzyme's really good at. And the place where they link up like puzzle pieces and match up and touch eventually, we call that the active site. They join together there. And right there where they join, that's where the chemical reaction will occur. Whatever activity needs to happen. So we've so far in biology class have talked about kinds of chemical reactions. We've talked about digestion, right? Digestion is a chemical reaction to break down food. We've also talked about photosynthesis. That's a chemical reaction. There are enzymes that help make that happen. And there's also cellular respiration. There are enzymes that work on different parts of the chemical reaction that make the reaction occur. Okay? So the enzyme is what's really great at making the chemical reaction go. And it's because it fits like puzzle pieces with whatever needs to have a reaction. Right? In this case, that thing's called the substrate. Kind of sounds like the word substance. The substance that needs to go through the reaction. Okay, hopefully you drew this picture. If not, pause before I keep going. Good. All right. So when we think about an enzyme, okay, the enzyme usually is drawn on the bottom, but not always. And I highly recommend you draw this picture as well, okay? So same idea, we have an enzyme and a substrate coming together at an active site, and here's the whole reaction. In the middle, they'll connect, right? And in this case, this enzyme's in charge of breaking something down, like digestion. And here we can see it's breaking two building blocks apart and forming products at the end of a chemical reaction. So if we thought of this as a chemical reaction, the left side of the chemical reaction, we always called them reactants, and on the right side we always called it products. This is the same exact thing we've been learning about. We're just learning about the itty bitty protein called an enzyme that does the whole process, okay? And the cool thing about enzymes that's amazing is, did anything happen to the enzyme that makes it so that it's broken or can't be used again? No, it's in perfect condition. Actually, an enzyme can do the same reaction over and over and over again. And that is important to how an enzyme functions. If something ends up ruining the shape of the enzyme, we have a big problem. Okay, so enzymes can be used over and over again. Right? Make sure you labeled the diagram in your notes. Pause if you need more time. Okay? So what do enzymes do? Okay, we've kind of been hinting at it. Let's summarize it. There are some enzymes that do what that picture showed, and they break down things. We know that a polymer, right, is a big molecule that has many units, polymer. And when they break them down, we break it down into its building blocks, and we call that monomers, 1-unit building blocks. And that's what enzymes do. They can break things down into building blocks. And when does that happen? In our digestive system especially. There's also enzymes that do the opposite process. They take building blocks, monomers, and they combine them and build polymers. So taking itty bitty Legos, right, and making a big castle, right? That idea is how we build muscle, right? So someone who wants to build muscle, they'll do exercise, they'll eat lots of protein, and your body, when it's eating the protein, it'll break them down. But some other enzymes pulling those pieces together to build bigger muscles, big polymers of muscles. So some enzymes break down, some enzymes build. Cool. Right? They are in charge of doing these important jobs in our bodies. And there's a lot of different enzymes, right? And I'm going to ask you to write down at least 2 of them. Okay, I'm going to go over more than 2, but all of them are going to end in the following 2 things. Either the end of the word will be ace or the end of the word will be in, okay? One example is amylase. This is found in, it's cut off your saliva. All right, so when you eat, let's say a cake, right, it starts to break down a little bit in your mouth. Without even chewing a little bit, you can taste the sugar and the sweetness. That's actually this amylase that's breaking it down. You can also, in your digestive system, break down lipids or fats because there's an enzyme called lipase that's breaking down those lipids. There's a enzyme in your stomach that breaks down proteins called protease. There's another one called pepsin. Again, remember that some of them end in ace and some of them end in in. You didn't need to write those all down. Maybe write 2 down, okay? Perfect. So just to kind of get a sense of what an enzyme does, here's an analogy. You don't have to write it down, but I think it's pretty cool, right? So I don't know about you. I worry about getting my hand cut when I cut bagels. So one day, Miss Clausen, she bought a bagel cutter, okay? And that's where you can kind of put bagels in to this device and it slices them up. Can I put a piece of cake in there easily? No. A bagel cutter, we're going to call it bagelase, is perfect at fitting in a specific substrate, right? They fit like puzzle pieces perfectly. And my enzyme, the bagel cutter, right, is going to cut them up. So is this enzyme in charge of building things or breaking things down? This one in our example is breaking things down. And I know this isn't a real enzyme, but it's the idea. So here's our reactants. We have our substrate. We have our bagel that's going to go into the place they meet, the active site, and our enzyme is going to do its job. When they're together, we call that an enzyme-substrate complex because they're complex together. They're together. My enzyme's starting to do its job, in this case, breaking things down, not building. And at the end, I have products. Right? I have 2 bagel halves I can make a delicious bagel sandwich with. And the cool thing is, do I still have my enzyme intact? Yeah. Can I still cut up more bagels? Yeah. Did it help me cut up the bagel faster than with just using a knife? Yes, actually, for Miss Clausen, for sure. So did it help me speed up the reaction of cutting up bagels? Yes. Enzymes speed up reactions. They have specific shapes that match up. Exactly. We have a little bit more to go through, okay? Almost there. So this is going to seem like a sudden change, okay? But we're going to review pH for a quick bit. When we talk about pH, it's a scale, goes from 0 to 14. When we look at the lower end, we call that acids or acidic. That's from 0 to about 6.99999, right? By the time you get to 7, that's neutral. That's our water, okay? And then if we get too high, okay, that's what we call basic. Being at either extreme is not the best. Certain things are better at being at different amounts. You can pause and kind of turn your head to the side and look at examples of different food or things that are these different pHs. One in particular is stomach acid is at a pH of 1. Right? We got Coke and Pepsi here between 2 and 3. Here at We have blood is a little bit around 7, approximately. We have baking soda and seawater and eggs are at 8, okay? Not too much stuff is really high pHs or basic. Our big contender here is bleach, which we use to clean. Pretty interesting, though. But why do we care about pH? You might be asking yourself, right? Well, enzymes are not so good at being in weird conditions. What happens to them, right? Remember, what was really important for an enzyme to be able to do its job? We called them like puzzle pieces. Its shape. Okay? So here's a normal enzyme reaction. Okay? An enzyme reaction will always have a peak. What do we call a peak in a graph? The highest point. We call that optimal, right? An enzyme is going to have an optimal condition that it likes the best, whether it's temperature or pH. Okay? This enzyme that's in green really seems to do best in around 1 or 2. That's kind of like stomach pH. Maybe this is an enzyme that lives in the stomach. But this enzyme, do you think it lives in the stomach? Does it do well? If its reaction rate is really low here, this enzyme probably doesn't live in the stomach because it wouldn't do a good job of reactions in the stomach. So enzymes do something called denaturing when they get at an unfavorable temperature, when they're far away from their optimum, right? Same thing with pH. If an enzyme is too far away from whatever its optimum pH is, okay, it will denature. Well, Clausen, what's denature? Sounds so weird. It's a really important vocab word, so circle it, star it, highlight it, okay? Denature means the enzyme loses its shape. So literally that perfect puzzle piece match, it's gotten all warped and funny looking. Can the substrate mix in with the active site if the shape is all messed up? Let's think about it. So what happens to the enzyme reaction when an enzyme loses its shape? So an enzyme that maybe looked like this, after it denatures, all right, the enzyme is going to suddenly look all like useless, like spaghetti. Can an enzyme substrate fit in anymore? Like a puzzle piece? No. No. So the enzyme with the wrong shape cannot do its puzzle piece match with the substrate, and it cannot perform its chemical reaction. This is a huge problem because if an enzyme denatures, the enzyme can't do its job anymore. It can't speed up chemical reactions. And this is really important because organisms need enzymes to do reactions fast. They are important for us to be able to survive. And we're going to talk about that more in class. What happens when our enzymes don't do their job because they're in a bad pH or a bad temperature. All right, so good job, you made it through one of the longer set of notes. I'm really proud of you. Make sure you jot down what denature means. You might want to even give an example picture here. Woo, I'm proud of you.